Hi everyone, welcome back to this week. Today I want to dive into a topic that was much discussed throughout the COVID vaccine rollout and recently revived by the Florida State Surgeon General. So let's take a look here. So now, in brief, in December 2023, the Surgeon General asked the FDA and the CDC if a DNA integration test was done on the two mRNA vaccine and received a less than satisfied response from the FDA. Now, so he thinks because there's no direct answer to the question, so it could be dangerous in theory, and let's put a stop to it. Now, just a word of caution here. Now, I'm going to address DNA integration mechanisms and with four different scenarios. And there is a lot of science in this video, and I will try my best to explain them clearly. So let's take a look here. So when does DNA integration occur naturally? Now let's begin by looking at two examples of when DNA integration occurs naturally with a virus infection. First, we have a vectoral virus infections like the human immunodeficiency virus or HIV. The HIV carries RNA as its genetic material, and after the viral particle enters the host cell, the RNA is released and reverse transcribed into viral DNA in the cell's cytoplasm, meaning outside of the nucleus. The DNA then forms a complex with viral integrase, an enzyme that can integrate the DNA into the host genome and along with other proteins. And this entire complex enters the host cell's nucleus where the human DNA is for later integration. Now, so from this example, we have several key moments we need identified. Number one, HIV RNA does not enter the cell nucleus, and RNA must be reverse transcribed into DNA before entering the nucleus, and the viral enzyme integrase is crucial for DNA integration. Okay, so now, the second example here is that we have a DNA virus infection like hepatitis B virus or HBV. Now, HBV carries partially double stranded DNA as its genetic material. And after the viral particle enters the host liver cell, the nucleocapsid or the coating of the partially double stranded DNA enters the cell's nucleus together. So the whole thing goes in together. But because the partially double-stranded DNA is a abnormality in the human cells, and the cells can recognize this abnormality and repair it to form a something called a closed circular DNA, and here abbreviated as cccDNA. This cccDNA is then transcribed into either messenger RNA and then also pre-genomic RNA. Now, this altogether subsequently formed viral proteins. But in some occasions, about 10% of the PG RNA can be reverse transcribed back into linear double-stranded DNA and move back into the nucleus. And here is the unique part of that. The HBV infections and other cell stress can break the human liver cell DNA and forming a gap here. Now, the science community currently propose that the liver cell would repair the gap with two DNA repair mechanisms. And the abbreviation here is the NHEJ or MMEJ, if you want to know the full name, which is called the non-homologous end joining and micro homology mediated end joining. So that's a mouthful of terms. However, if and when HBV linear double-stranded DNA is present, that this piece of virus DNA would get integrated into the human DNA. Now, and the implication is that it could lead to further liver pathology, such as like cirrhosis, hardening of the liver, further exacerbations of hepatitis, and also cancer development because there is a mutation in the liver cell genome there. All right. So I know there's a lot of deep science here. So let's regroup a little bit with a little bit more simple language. All right. So what does that mean? Let me pull up some slide as well. All right. So here are several points I want you to take away with those two natural DNA integration examples. Number one is that viral DNA can integrate into human cells and be potentially cancerous. And number two, viral DNA can be integrated by a viral enzyme called integrase. And number three, viral DNA can also be integrated by human cell DNA repair mechanisms where there is a breakage formed from infection or other stress. And number four, viral DNA needs to be carried into the nucleus through a specific mechanisms, okay, and depending on what the virus is. And last but not least, viral DNA integrations happens when the virus is actively replicating, and that is the key, meaning there is a lot of viral DNA present. And this quantity factor matters because DNA damage in cells or mutations happens, often, but not all DNA damage causes cancer. It is when there is way too much DNA damage that is beyond what the cell can repair, it then becomes cancerous. So let's keep these five facts in mind because they are important for our next few discussions here. All right, so now the second question here, or second scenario, is that does SARS-CoV-2 genetic material get integrated into the human cell? Now, this is a very controversial topic, and the first related paper was published in a journal called PNAS back in May 2021. Now, this study used cell cultures and showed that on rare occasions, an enzyme in human cells may copy the viral sequences into the DNA and slip them into our chromosome. So you may ask how. Now it turned out that let's look at the bigger screen. It turned out that 17% of the human genome consists of sequences called line 1, which is an artifact of ancient infection by old retrovirus throughout the evolutionary of human or related species. So line 1 is a type of transposable elements that can move or jump or transpose within the human genome, and it is often referred to something as retrotransposons because its mechanism involves a reverse transcription of an RNA and then back into an integrations there. So the retrotransposition process of line 1 involves several steps. First, the line 1 DNA is transcribed into RNA by the whole cell machinery, and then the RNA is then reverse transcribed, here shown in the figure, back into DNA by the line 1 encoded reverse transcriptase. And then the newly formed DNA is then inserted back into the new location in the genome. So that's a lot of science again. So what does that mean in a simple way to say it? Is that, long story short, this study showed that it is plausible, all right, for SARS-CoV-2 virus RNA to be reverse transcribed and integrated into whole cell DNA in a cell culture experiment. Now, however, there is no new evidence showing that this could happen in human body. Now, that brings us to the next question here. All right. So, which is, Could vaccine RNA or messenger RNA integrate into human cell DNA? I think a lot of people were asking those back in the time, and let's a little bit have a little bit regroup and review of the current information. So, the same group of the researchers that did the study about the SARS-CoV-2 viral RNA integration into cell culture DNA did a follow-up study using a similar methodology. Now, this study was published in early 2023, right? So, but however, for whatever reason, the research group could not get access to the actual vaccine RNA, which is packaged in the lipid nanoparticle. That instead, they created a model of vaccine injection, inserting a bit of SARS-CoV-2 messenger RNA into cells through a process called transfection. This transfection process is a non-infection delivery of genetic material into cells. And the study showed that introducing SARS-CoV-2 messenger RNA through transfection did not result in genomic integration as observed in natural infection. And infection induces a significant production of viral RNA, just like other viral examples that we talked about, and trigger cellular inflammation, so some type of a stress, and elevating or increasing the activity of these reverse transcription machinery, the line one mechanism that we just talked about. And this highlighted that stress in cells is crucial to genomic integration. So, in contrast to transfection, this does not induce inflammation or stress, and they find no indication that it lead to viral genomic integration through the line one mechanisms in these normal cells. All right, so that's a lot of information. So basically, they've repeated that experiment that they did earlier with infection, and they show kind of indirectly show that with just the messenger RNA putting it into the cell, there is no known mechanisms for it to be turned into DNA and brought into the nucleus because the cell is not under stress based on their experimental model. Now, and I know there is a different question here because what the Surgeon General brought up was something called DNA fragment, right? So, what about these DNA fragments that was left in the vaccine? So, does all these things come from? All right, so in early 2023, the plasmid DNA contamination theory surfaced, and the theory originated from a preprint study, and which is still in preprint, showing that the two messenger RNA vaccines had a nanogram to a microgram of fragmented plasmid double-stranded DNA per vaccine dose. And they claim that this amount would be more than the acceptable safe levels of plasmid DNA that are left over from the manufacturing process. Now, in addition to this preprint, a professor, Dr. Philip Burkhoff, professor of cancer molecular genetics at the University of South Carolina, testified to the South Carolina State House and brought up the concern that those plasmid DNA fragments contaminates may not be as biologically inactive as the regulatory agency and the vaccine manufacturers claimed. So this one went on, there's videos on YouTube. We could watch that actually. So now the problem is that the origin of these plasmid DNA is actually a leftover from the industrialized large-scale vaccine production. But the Pfizer mRNA vaccine used in early clinical trials used a polymerase chain reactions or PCR process to make the DNA that code for the messenger RNA and then be put into the particle and packaged into the vaccine, a so-called process one. But when you are trying to scaling up the production to make millions of and billions of doses, the manufacturer had to use something called a plasmid, which is a small circular piece of DNA that put into a bacteria cells, and this is called a process two. Now, because it is much cheaper and faster to produce a messenger RNA this way, compared to using the PCR format that they used in a small amount during early clinical trials. Now, the documents it's actually showing that the regulatory documents showing that the process one and two, they claim to have the same product. Okay, and the FDA is aware of that before even they authorize the emergency use. All right, just to be clear on this side. Now, the disadvantage of using plasmid to generate these messenger RNA is that the end product needs to be purified to remove these residual plasmid DNA. Now, at this point, usually these plasmid DNA would be fragmented already, okay, because of the extraction process and all these technical things that I don't want to go too much into this video. Now, all right. So, does leftover plasmid DNA matters? All right. Does it matter? Is it new? Now, it turned out that this is not something new. Now, according to a conference report led by several world-renowned vaccine experts, okay, say that basically the harmful risk from residual DNA has been debated for more than 50 years, and the FDA does have industrial guidance to limit the amount of residual DNA left over from cells that are used to make a vaccine. And also, these tests all have to be done by independent labs. So it is not just the manufacturer doing these testings. So, in other words, yes, there are plasmid DNA fragments in messenger RNA vaccines, but they are also present in many other vaccines dated before messenger RNA vaccines. So let's be clear on that. Now, but there's also another layer to the story, right? If we read the Surgeon General's letter, which is something called the SV40, right? So what is SV40? So SV40 is a monkey virus. It has been shown to cause cancers in rodents, and polio vaccines used in the late 1950s and early 1960s were contaminated with the virus, the SV40 monkey virus, which was present in the monkey kidney cells that was used to grow that vaccine at the time. Now, however, over the decades, extensive long-term research did not see people who received the contaminated polio vaccine had a higher risk of cancer when compared to those who did not have the polio vaccine. However, on the other hand, that based on the preprint study, that it is confirmed that a small DNA sequence uh, from the SV40, it's called a promoter sequence, is present in the entire plasmid construct that is used to make the messenger RNA. It is important to know that it is not the entire SV40 virus is in that plasmid. And also, there's another very important antigen called the SV40 40T antigen. Now, this antigen is known to cause cancer, and this is also not this is also not present in the plasmid based on these experiments done by people that are not from the manufacturer. Okay. Why does SV40 matters, and is it really some arguable point? Okay, so it turns out that there is a research group from the University of Rochester Medical Center. It is called the Dean Lab. Okay, they specialized in studying SV40 promoters, or enhancers. But it concluded that these SV40 promoters or enhancers, along with other proteins, are required for plasmid DNA to enter the nucleus. Now, notice that the report showed that the entire circular plasmid DNA enters the nucleus, and it did not show the entry of fragmented DNA or any DNA integration into the cell's native DNA. All right. So, well, there was a lot of science here. I believe it is time to regroup a little bit too. So, in human words, I know I didn't speak human earlier. Now, let's speak in human words. If you are staying until this point, messenger RNA vaccines, okay, have a little bit of fragmented plasmid DNA, and that is within regulatory guidelines according to the agencies. All right. Now, some researchers believe that, the lipid nanoparticle that is, used to package these messenger RNA, that at the same time also package these fragmented DNA, makes that the little bit of DNA more problematic and can enter the cells more efficiently. Now, yes, SV40, okay, some of these promoter sequences can play a role in bringing the entire plasmid DNA into the nucleus, uh, but it also needs other protein for it to do the job. So it's not just that promoter, it's not just the DNA, there's also other things in that complex to package to bring that plasmid DNA into the nucleus. And there's no research currently showing, linking the SV40 promoters and the fragments of DNA entering the nucleus. Notice that the Rochester group, okay, is showing the entire plasmid DNA, right? Versus what we find in the vaccines, not just the mRNA vaccine and other vaccine as well, are fragmented DNA. So small pieces, right? There's a difference there. So, so there's really no evidence currently showing, once this fragments of DNA enters the cells or even enter the nucleus, that could integrate into whole cell DNA, right? So there's no evidence currently. Now, however, now there are some, I mentioned earlier on some natural phenomena that, when the cells are under stress, there is gap forming, there's a possibility, there's some theoretical mechanisms that could integrate fragments of DNA into whole cell DNA, okay, when the cells are under stress. What is important to note that is that the amount of DNA that would be integrated matters. The case that we saw with natural infection and also with the experimental group, that the experiment within the cell culture that did by one group, is when cells is under stress and there is an integration happens, and also when there is an infection, when there is a large amount of DNA that is present. So now, so a minor amount, okay, a small amount of integrations, of DNA materials into a genome does not necessarily always lead to cancerous development, because there is one very important system in our body, which is the immune system. The immune systems are very efficient in recognizing these small changes and clear the mutated cells when the amount is relatively low. All right. And when we look at all the currently known science, when the Florida State General Surgeon General is concerned, what he is concerned about currently, what he stated in his letter, is a hypothesis at this point. All right. Now, for now, integrations of fragmented DNA from vaccines and that causes cancers is quite improbable based on what is available there, meaning it is unlikely, but at the same time, it is not proven to be 100% impossible. So where that lead us? Well, to quote Sherlock Holmes a little bit, now one can only find the truth when all impossible is eliminated. All right. So currently, this is should be an open scientific debate. Unfortunately, the science of messenger RNA technology or mRNA technology is now highly politicized and highly commercialized as well. And everyday good science can no longer be done easily in this realm, and a lot of things are protected by these, manufacturers. So if if the mRNA technology is as safe as the manufacturer and the regulatory agency claims, then I think they should prove it with evidence. Now, the manufacturers have the resources to do those experiments. And at the same time, I think when academic expert propose concerns about the technology or hypothesis about certain things, they should also be given with the resources to test their theory. And this is the way of science. But when or if we will ever see this, that is an even bigger question. Well, okay, so that's a lot of science, very long presentation. I look at the clock, it is 20 some minutes there. Now that is all for this week. I know this video is super deep in science, and I know I will receive comments saying that they don't know what I'm talking about. But to those who watched it to the end, I wholeheartedly appreciate it. Now, thank you very much for watching again, and I hope to see you in my next video again. All right, so please take good care, have a good rest of the day or night, and see you next time. Bye.